So let's move on to duplications. So with duplications, the genetic material just gets duplicated. It's a repeated segment of a chromosome. This can be due to unequal crossing over, which we'll show in the next slide, or it can be through some kind of replication error, and results in what we call gene redundancy. Now, it can produce a variation in phenotype and will also give those compensation loops that are similar to deletions when the heterozygotes pair during meiosis. This is actually can be a good thing. It's an important source of genetic variability. So this is showing you how, if you have unequal crossing over occur here, so instead of it being the crossover occurring in the exact same region, it's occurring here between B and C on the blue chromosome and C and D on the orange chromosome. And so you end up, in the process, you end up with a duplication. So here you can see chromosomes 1 and 4 are normal. Chromosome 2 ends up with a deletion, so this is also a way that you can get deletions. Chromosome 3 has a duplication. So the resulting gametes will be abnormal if they contain either chromosome 2 or 3. Now, duplication allows you to make extra copies of genes. And this is a good thing because in some cases we need more copies because we need more gene product. The ribosomal RNA is a good example of that. The ribosomal RNA is copied multiple times. You have can have hundreds of copies of the gene for the ribosomal RNA to make ribosomes, and it's just a supply-demand kind of thing. You need a lot of ribosomal RNA so you can make a lot of ribosomes so that you can make a lot of proteins. And if you only have one copy of these genes, it would be inadequate for the cell. And this is called gene redundancy. Now, sometimes it can be a bad thing. In Drosophila, the bar mutation, so the bar eye phenotype is the slit-like shape of the eye, and this results from duplication, and it's an inherited mutation. If the flies have 2 copies on one of the X chromosomes for this gene, then they're heterozygous and they look like this fly here. So you can tell it's slightly skinnier than normal, but not as skinny as if they are homozygous for the condition where they have 2 copies on both X chromosomes. Then they look like this fly here. So gene duplication is actually really important for genetic variation because what happens is if you have an essential gene and it gets copied, now that gives you the opportunity to play around with that second copy, and you still have the original copy intact to carry out the function. So if you have, say, gene A and B, with B being if you have 2 genes present, gene A and gene B, and they both get duplicated, the original gene can continue to do the job that it was meant to do. The duplicated gene, but is now free to mutate and can become something else. If it ends up being bad, then no worries because gene A is still carrying out the function, but it can end up being a change that leads to the organism giving it some kind of advantage in its environment. And so that's what evolution is all about. So you can gain genes with different roles, leading to different phenotypes, can even result in a different species evolving and can give that species an advantage in its environment. So from this type of change that occurs over time, we end up with something called gene families. And so these are groups of genes whose products perform same or similar functions, but they differ, in different species. One example is this gene called sRGAP2, present in primates. It's involved in brain development, and we have at least 4 different copies, similar copies, and so they're just designated A through D. Primates, non-human primates like apes and monkeys and chimpanzees and whatnot, they have only one copy. And it appears that the duplications, when they emerge, coincide with the emergence of our lineage from primates. So by making copies of this gene and then those copies getting changed, it actually developed our species and allowed us to have extended evolutionary brain function.